drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. Telling everyone a little bit about what you do today. What do you do for work, and what does that involve?
Well, at the highest level, what I would like to do is to bring true preventive medicine to the society, and that is to essentially empower individuals to live the same life that people in the blue zones do, which is they live into their 80s and 90s very healthy. They have physical health, mental health, cognitive health, and immune health well into 80s and 90s. And nature has already figured out how to do that for some people, those that are born there. Sadly, we don't have those kinds of lives in modern society in the developed countries, but we have the science to figure out how nature has enabled this kind of a long health span, and then with testing and AI, enable individuals to live that kind of a life. That's at the highest level. And then how I have gone about doing that, well, that's a 15-year story. So I'll just give a summary that about 15 years ago, I basically changed my scientific career. I was a government scientist, and I was mostly working on the drug development and drug discovery. And I realized that I actually wanted to do this preventive medicine strategy. So I changed my scientific focus in 2010 and started developing a systems biology view of the human body. And a systems biology view means that there isn't just one factor that affects our health. And so there are people who are geneticists and they focus on just the genetic contributions. There are people who believe that the gut microbiome is the root cause of all diseases. There are people who think that heavy metals are the root cause of all diseases, and so on. But the real truth is that for a population, there are many, many factors. And even for individuals, there are likely many factors that contribute to our health and disease. And we really have to look at the entire human physiology. And what I realized back then was that we needed to have a very, very solid molecular test that profiles that physiology. And at that time, I decided that I was going to pursue RNA and not DNA or other molecules in the body as sort of the best molecule to characterize our physiology. And so today, for example, what we do with our stool, blood, and saliva test is that we look for over 100,000 different kinds of molecules that maintain our physiology. And so it took about 6 years as a government scientist to develop this foundational technology. And then in 2016, I left the government scientific position and we spun out Viome with Guru Banavar and Naveen Jain. And now over the last 8.5 years, we've been running the technology at the company and scaling it and adding a lot of AI and adding a lot of knowledge and so on.
Mm-hmm. So when you talk about, so you spun things out and did this company based on the research you were doing at the government, you have an academic background, and you're talking about preventative medicine and systems biology. What sort of led you to that transition? Why did you not stay on the government research track you were on? Were you not able to do preventative medicine stuff in that context, and so you needed to switch out of it? Like, how did that sort of transition happen, and what was like propelling you?
Yeah, so when I initially envisioned this whole platform, I did not make a decision as to what kind of an entity was going to actually exploit this sort of a platform. And as an academic, my natural sort of tendency was, well, I'm just going to start a nonprofit institute, right? Mm-hmm, mm-hmm. But as years went on and I did more research and I did more talking, I realized that the only sustainable way that this can be brought to the humanity was a for-profit business. And the reason is because these nonprofit institutes and foundations, they spend most of their time actually, essentially begging for money every year, right?
Yeah.
You have to
Yeah.
write proposals, it's just scaling becomes a problem at every possible day. And so I thought, let's make it a for-profit so that we can actually scale more rapidly, have lean operations, and get some money in so that it can be self-sustaining and self-sustainable. And that's what we chose. And I don't regret that decision, but could we have gone as a nonprofit and applied for SBIRs and applied for other government grants? Probably. It's just, I think that this was the most efficient way to do it.
But it sounds like also that you were doing what we might call more traditional drug development stuff before you went this direction. What is sort of the difference between preventative medicine and traditional drug development, and how did you sort of think about that?
Yeah, I mean that's a fundamental question. So preventive medicine is figuring out why is it that nobody on a Greek island who lives to their 90s develops inflammatory bowel disease, right? Nobody. But their children who move to the city develop IBD, right? So it's not genetics. They're in the same country, same weather, similar foods, but all of a sudden their children develop all these chronic diseases. So what is it? It's not magic. It's not extraterrestrial force. It's not bad luck. It's simply there's a breakdown in some kind of a physiological pattern. And clearly, people who live the traditional way on the Greek islands, they have all the molecules in their body necessary to prevent inflammatory bowel disease, whereas their children have lost that ability and now they develop the disease. And so the concept is, can we find out what those molecules are and provide them to the children so that they never develop IBD? That's preventive medicine. Reactive medicine, which we have today, is, well, let's wait until that person develops IBD. And now let's basically find a place in their body where we can sort of stop that from happening with all the other consequences that come with it. And so, for example, for IBD, that I chose, there are pharmaceuticals that can treat it, but they're not super successful, and they have very heavy toll in terms of side effects and financial toll. And so, why not just figure out what is it that keeps you healthy instead of react to a disease?
And when you say that, so in this example of IBD for people from the Greek islands, it's not genetic because you're talking about one generation of difference here. You start seeing it in the children of people that don't have it. The genetics are not substantially different. They're living in the same climate. They're living in at least a highly similar culture and eating, if not identical, very similar foods, but they're missing molecules, you said, that prevent IBD. How do we know they're missing them? And if everything is very similar from one generation to the next, what's actually missing? Is it something in the food or what?
Yeah. So that is really the question, what is missing? And what is going to be missing is probably many different molecules, not just one or two. Any chronic disease is going to be very complex. And here's where I think it's very important for people to understand how resilient our physiology is. Think of the human body, and we can sort of follow this thread throughout the conversation. Think of the human body as a very complex machine, such as an airliner, like an Airbus A380. And that Airbus airplane has a lot of different systems that enable it to fly very fast, very long distances, but it also has many redundant systems so that if one screw falls out somewhere, the plane doesn't crash, right? It has multiple redundant systems. And so you as a passenger sitting on a plane, flying over the North Atlantic, if a screw falls somewhere or some electronic system fails, you're not going to notice anything. There's not going to be any symptoms. And the reason is that all these redundant symptoms pick up. But when that plane lands, if you don't fix the problem, you just keep flying, eventually enough of these redundant systems will break, and then you'll have a symptom of a plane crash. That's a very good metaphor for the human body, where we have redundant systems for maintaining our physiological health, but over time they start to break. And those are pre-symptomatic. You don't get to see those with symptoms. You don't feel any difference, but we can measure those. But when enough of them accumulate, then you become sick. You develop a chronic disease or cancer.
Mhm.
And so it's going to be very complex to figure out all of the factors that contribute to every chronic disease. So, for example, with IBD, we have identified, when I say we, the scientific community in general, not just we at Viome, we have identified both microbial and human components that either independently or often together combine in order to cause IBD. And so in this particular case, I'll give you a couple of things. And so in one particular microbiome only specific case, what was recently discovered was a bacterium. It's a Streptococcus salivarius, which is typically living in our mouth, but it can travel and live actually in the gut. It produces a peptide called nisin, and this peptide inhibits Fusobacterium nucleatum, which is very pro-inflammatory. And there are other cases where you have microorganisms that normally process bile acids that leak out of the small intestine into the large intestine. They process them into secondary bile acids, which do not cause inflammation. So bile acids, think of bile acids as literally detergents. They're dissolving fats in your food in order to help digest. And they're almost all of them are supposed to be reabsorbed in the small intestine back into the bloodstream. But some of them do go through and enter the colon. And that's okay, because normally those old people who live on an island somewhere, they still have the microbes to convert the primary bile acids into secondary bile acids and basically render them not inflammatory. But if you've lost that microbe because you've taken too many antibiotics, you've had diarrhea too many times, you've eaten too many preservatives, now the primary bile acids enter the colon and they're detergents. They're stripping away the surface of your colon, and now you can have inflammation. So those are all sort of microbial root causes, but there are human root causes, root causes, but human factors that contribute to IBD, where we have inflammatory pathways that can recognize the presence of pathogens where the inflammation starts. And that's what basically triggers the inflammatory process in IBD. And that process is supposed to slow down and stop at some point in time. But if the inflammatory signals from the gut microbiome continue, then the inflammation continues. And if the person has a genetic predisposition to not calm down inflammation so easily, then that inflammation can really inflame the whole gut. And so these signals are produced by the gut microbiome to start with, and they're called lipopolysaccharides. So there are chemicals produced by the gut microbiome called lipopolysaccharides. Some of them will activate our immune system. Some of them will prevent that activation. So it's the balance of those two that decides, okay, my immune system is going to get activated. But in most people, the immune system activates a little bit and then calms down and says there's nothing to see here. But in some people, it continues to be activated, and then it causes an inflammatory reaction via these receptors called TLR4 and downstream effector molecules such as IL-1 beta and so on. And so just one disease, such as IBD, is multifactorial. It includes what foods you're eating, what microbiome you have, what microbiome you're missing that you should have, and what is your genetic predisposition. That entire combination is going to determine whether you have IBD or not.
So some people might just have a genetic constitution that makes their tendency to gut inflammation a little bit higher. Other people might naturally house certain bacteria more readily than others. Other people might lose certain bacteria because they had antibiotics as a child, or because they started or stopped eating something like a processed food that contained effectively antibiotics, preservatives, that got rid of that species. And like the one example you gave is, there are factors that the microbes produce or factors that our own bodies produce that can be pro-inflammatory in a context-dependent fashion. So maybe it's pro-inflammatory when it's this primary bile acid that our body creates. But if you have the correct, quote unquote, microbe in your gut, it's then processed into something that's not inflammatory. And so all of these things interact, and it can result in IBD in certain individuals.
And this is what we know. We don't know what we don't know. In our own internal data at Viome, we have characterized all IBD patients into 4 very different groups at the molecular level. And they have some commonalities, but they have some very big differences. that we do not understand biologically what that means.
Yeah.
And so even a disease that people call IBD, which is like a uniform disease, well, if you just take just ulcerative colitis, that is one disease because there's also Crohn's disease. So if we study just ulcerative colitis, which we have focused on, there are 4 subtypes that we don't quite understand, what all the factors are. So for each one of these diseases, it's going to be multifactorial, and the factors that play out the disease in each person are going to be different. That's the thing.
Yeah.
So you can basically think, this is the way I think of every disease. Let's say there are tens of factors that in a population are involved in promoting that disease. But you only need a few of those in order to actually induce the start of the disease, to start the disease. And in any one person, that combination of different factors will be different.
Yeah.
And so you have this very complex landscape that you have to manage in so that you can have a true preventive medicine.
Yeah. And that would imply, so basically what you're saying is, we can call one symptomatic manifestation, a disease like IBD or whatever, but you and I could present with similar symptoms and get diagnosed with IBD or something similar, and yet the pathophysiology is very different. The origins of how that got started are completely different or largely different between you and me, which would therefore imply that the treatment that works for me is likely not going to work for you.
That's correct. And the prevention strategy is going to be very different. And so I'd like to tie this. I really like this metaphoric comparison with Airbus A380. And so there are many reasons why a complex airplane like that is down, meaning that it is unable to fly. Many reasons, right? Because there are so many systems in that airplane. And so when someone says, "Oh, engine 4 is disabled," right? That would be like saying this plane has IBD. But wait a minute, engine 4 is not like one block that's either working or not working. It has thousands of components in it, and any number of combinations of faulty components can lead to that engine not working, right? And so we think the way I think about it is that we have many, many different kinds of diseases, many overlap, and each one of those, again, multifactorial, and then in each person, there's different factors that can go wrong. So what we need to do is exactly what we've done for airplanes, which is understand all the parts that contribute to a healthy, functional airplane, have a yearly or quarterly testing of every single part that contributes to that healthy airplane. And then every time that we find a faulty part, we fix it immediately. We don't say, well, it's just one part, the engine is not broken, we can wait until a few more parts break down. Because if we wait for a few more parts to break down, now the plane crashes. And in that world, humanity has decided we do not tolerate planes crashing for maintenance reasons. We're not going to tolerate 400 people dying because we know how to prevent it. On the human health side, we as humanity have decided it's okay to wait until you're sick and dying because, well, that's how healthcare makes their money. And so that's the foundational sort of mindset that we have to change, is that we can, in fact, completely bring preventive maintenance to the human body. We just have to do it. And the main challenge has been that no one has basically designed a business model where people are willing to pay for preventive maintenance. And I think that that's really the key. There are now many ways in which you can reduce the risk of chronic diseases and cancers. They are very well known. But how do you get someone to not eat something that they love?
Right.
Is a big problem. How do you get people to exercise when they don't like to exercise? That is the main problem, I think, with preventive medicine, is that people actually have to do something about it, just like what we're doing with airliners. The airliner is not going to fix itself, and it's not going to be healthy forever. There's an army of people actually doing work to maintain that. And so it takes work. So that's really the biggest challenge, I would say. And that challenge has always been because, hey, I'm not going to stop eating steak because my uncle ate steak all his life and he lived to 95. So you can't tell me that eating steak is bad, right? We have to bring data to every single individual and say, this is why you should not eat steak, because for you, steak has these micronutrients that when your microbiome processes, it causes cardiovascular disease, and your risk of a heart attack is very high. Your uncle doesn't matter because he had a very different microbiome than you. So we have to bring people the data.
Yeah, I want to there's a lot of things we'll get to, but I want you to unpack more examples like you just gave where one food can be good for one person or bad for the other person. Like, so a lot of times, even not just among lay people, but among physicians and health experts of different types, people are in this trap of thinking of foods as good for you or bad for you. Broccoli is good for you, depending on who you ask. Steak is good or bad for you, depending on who you ask. What are some examples where a food could be good or nutritious for one person, but not the other? And then what do we know about the biological basis for what accounts for that?
Yeah, I think that's a key aspect of our nutritional platform, which we call Nutrition 2.0. And so I encourage people to just Google Nutrition 2.0 Viome, and they can find an article that we wrote about that. But basically, that is really one of the biggest takeaways from this entire conversation. Why is there so much conflicting information about nutrition today? If you like to eat something, you can definitely find evidence that that something is good for you.
Yeah. You can justify anything if you just know how to look for the evidence. Yeah.
And if you don't like to eat something, you can definitely find evidence that that's bad for you. And so the reason for that is that the vast majority of studies, nutritional studies out there, are epidemiological. That means they're done on a population, and they're asking the wrong question. And the question is coffee good for all humans, or is coffee bad for all humans? That is the question. The real answer is that coffee is good for some humans and coffee is bad for other humans. But that's not the question they're asking. They're asking the wrong question. And if you ask the wrong question, you can never get the correct answer. And so when you ask, are eggs good for you or bad for you, you can never get the right answer. Okay, so let's talk about examples. So I'm not on the clinical nutrition side, so you can have a 2-hour talk with our clinical nutrition team about all of the examples, but the ones I've seen are, for example, the ones I like in particular would be something like oxalates. So oxalates are pretty famous plant toxins.
Yep. And spinach and other things.
Yeah. So oxalates are present in some legumes, in spinach a lot. And so this is a controversial topic. Traditionally, again, our microbiome, like our ancestral microbiome, had plenty of bacteria to feed on the oxalates that we consumed from plants. We co-evolved with bacteria. They were basically symbiotic with us, and they would be like, hey, you're consuming oxalates. You don't have a way to break them down. They can make you sick, but I can. And so those bacteria started living with us who knows how many hundreds of thousands of years ago. And so we consume plant foods where we extract good stuff, and the bacteria extract some other good stuff. But now with modern life, where we are taking too many antibiotics, too many food preservatives, eating a clean life, and we should definitely talk about all those topics. Now, more and more people are starting to lose their ability to process oxalates. And so if your microbiome has lost the ability to process oxalates, then you should minimize those high oxalate containing foods. It doesn't mean you should stop eating all the foods that have oxalates, because that would be very detrimental for you, but just minimize. And I like this particular example because it applies to me. I've had a good oxalate score since we started it many years ago until about summer of 2024, when my oxalate score went from optimal to suboptimal. It wasn't bad, just suboptimal.
Yeah. Meaning you had the ability, you had a microbiome that gave you the ability to tolerate reasonably high levels of oxalates, and then for some reason it went down.
Any level, no limitation on anything. I had a microbiome that was very highly capable of processing oxalates in the foods, and so I had no restriction on that. And soybeans were my superfood, black beans were my superfood. And I am mostly vegan, and so I used to eat soybeans 3 meals a day, every single day. But last summer, the test said, oh, your oxalate score is now suboptimal. And so the computers automatically said, well, let's look at all the foods. And so what the computers did, this is all algorithms, AI algorithms, and the computers know exactly how much oxalate there is in every food. Oxalate said, go ahead and reduce soybeans and black beans because they're the highest of oxalates. Our computers have every single detail about every food in terms of micronutrients. But guess what? Chickpeas and lentils are much lower in oxalates. So now move those and eat more of those, right? And so I was at first kind of sad, frustrated, devastated about this because I was so used to, I had developed basically a full menu of foods where I was dependent on tofu. But I said, hey, this is my biology. I better listen to this test. And so that's the data that made me say, well, this is potentially serious. Let's do something about it. So I just spent one weekend, literally one weekend, reading, exploring, and experimenting in the kitchen. And I essentially devised a tofu recipe from chickpeas and lentils. And so all of a sudden, after that weekend, I started making tofu not from soybeans, but from chickpeas and lentils. And not only is that now better for me because it has lower oxalate content, but these tofus actually taste better, they're environmentally better, and they're better whole foods than soybean tofu that you buy at the grocery store. And we can dive into those details if there's interest. But
so for this example that you're giving for yourself, were you experiencing any problems, or was this the type of thing where something changed and the test was essentially telling you you better back off the oxalates, otherwise you're going to be probably having kidney stone issues later on?
Exactly. So no, nothing was happening to me. And so this is one of those things where you can prevent disease years or decades before it sets in because you're understanding how your physiology is changing. And no, you don't develop kidney stones overnight. They take years or decades to develop, depending on many factors. But these are the kinds of test results that can warn you about that, and you can change your diet before the disease ever sets in.
Yeah. And I mean, tell me if this is true. It sounds like there was no obvious trigger for this. Like you weren't in the hospital being pumped full of antibiotics. Something just changed. And I would imagine that happens to a lot of people where it's not obvious how their lifestyle is changing, but something must change. And something that would otherwise fly under the radar was made apparent to you.
Yeah, I mean, exactly. So first of all, it wasn't a dramatic change. I didn't just lose all of a sudden the ability to process oxalates. It just diminished. I am getting older, so that's one thing.
Yep.
And the second thing, it's a crowded space down there. You know, there's about 1,200 species of microorganisms, and they're all trying to survive. And they're all, you know, it's like that metaphor putting 6 rats in a bucket and giving them only food for 4. You know, there's always diminishing food supply. So yeah, everything you said is exactly right. There's nothing obvious, nothing that I've done. I didn't take antibiotics or anything, and I didn't certainly kill the ability of my microbes to process. It just diminished their ability to do so. And this happens with aging.
And so can you start to give us a sense for like the products that you offer at Viome? How do they work? What are some of the major components in terms of what's being measured and how it's being measured and how it is more comprehensive than, say, just getting like a blood test?
Well, we're measuring a very significant part of human physiology, and I would say that it's complementary to a lot of other things. You know, I have my sort of, and I can tell you what my recommendations would be for anyone. So let me just very plainly explain what we offer at Viome currently. So we offer test kits which measure stool, blood, and or saliva. I would obviously prefer that every customer measures all 3 of those, blood, stool, and saliva samples. And we currently look for about 100,000 different things in those or physiological biomarkers in those 3 samples. And we actually detect and quantify about 24,000 of them. And so when you go get your metabolic panel done or lipid panel, you get a few biomarkers. We report 24,000 markers from every single person. That's a very deep, it's sort of like, again, going back to the Airbus A380, a metabolic panel would say, this plane has 4 engines, it has a fuselage, it has 2 wings, and it has a tail. Looks good, right? Ready to fly. Well, not so fast, right? But our test, we're going to send out, 100 people to open up every panel, to check every screw, check every connection, check every cable, check every electrical supply, check 24,000 data points to look for, are there anomalies with those? And we can dive deeper into exactly what we do, but we can infer some some lipid and some metabolic biomarkers, but for the most part, they're complementary. And so we test and we measure those 24,000 markers, and then based on which ones are out of order, meaning they're either, so these are basically, think of them as biochemical reactions. And I can dive deeper what a biochemical reaction means, but it's basically processing of some micronutrient from your food into some other chemical that the microbe doesn't need anymore. It's like micro poop, basically. And during that processing, the microbes harvest the energy from that food, and that's how they survive in our intestines. And so we measure about 24,000 of those chemical reactions, and I quantify them. And so we know which ones are too fast and which ones are too slow. And typically, if they're either too fast or too slow, that means that something is not right. And so we want to slow down the too fast ones, and we want to accelerate the slow ones. And how do we do that? I'll give a very, very specific example that people are probably very familiar with, which is, how do we make beer?
Okay.
We make beer by providing the substrate for a microorganism. So the substrate is the sugars that are found in barley. The microorganism is the brewer's yeast, or Saccharomyces cerevisiae, and the product is ethanol. And so if you want to make beer, you have to have barley and yeast. If you put those same yeast on lettuce, you're not getting your beer. All right. If you put that same yeast on a piece of steak, you're not getting your beer.
Yep. Specific substrates facilitate the growth of specific microbes, which is why our diets affect the microbiome composition.
Exactly. And the most important part is that metabolic output, the ethanol, the alcohol that the yeast produce. That ethanol in general is bad, but let's say there's a optimal amount of that ethanol that's produced. Let's actually switch from ethanol to butyrate, because that's actually physiologically relevant. But think of, there's a bacterium in your gut microbiome that when it consumes a very specific polysaccharide, it will produce butyrate. And if you have the right amount of butyrate, it is great. It is feeding your intestinal cells. It is providing all kinds of signals for your immune system. It is a very healthy product at the right level. If your bacteria producing too little of it, that's not good for you. If they're producing too much of it, it's also not good for you.
Yeah, I would imagine that's probably a pretty general principle, right? Like many molecules in the body, you want at some Goldilocks level.
Exactly. That's exactly what it is. It's just like cholesterol. Yeah, too high of a cholesterol is bad, but too low of a cholesterol is also as bad. And sugar, too high blood sugar is bad. Too low of a blood sugar, you're dead. So even these things that we think of as, we got to get it lower and lower, that's still lower within the healthy range, but there is too low.
And that seems like an important thing too, because a lot of medicine, maybe inadvertently to some extent, but there's a lot of focus out there on lowering the so-called bad markers rather than thinking about the optimal range.
Optimizing them, yeah, exactly. And we're trying to optimize everything. Okay, so let's go back to our example. You have a microorganism that can process a specific polysaccharide that you consume and produce butyrate for you. Most people have many such bacteria, many different species. And they consume different polysaccharides and they produce butyrate at different rates. And so when we quantify your butyrate production, we know what level is healthy.
So you're not just measuring if people have butyrate or not, you're measuring how much and how quickly it's being made.
We measure the biochemical activities of the microbes for production of butyrate. And so we don't just say you have Faecalibacterium prausnitzii, which is capable of producing butyrate, but it doesn't have to produce butyrate, meaning that the presence of a microorganism doesn't say anything about its biochemical activity. Because, like I said, if you put brewer's yeast on a piece of lettuce, you're not making beer. And so we're measuring exactly the biochemical activity for a specific chemical reaction for about tens of thousands of those. And so now if you have just the right amount of butyrate, that's great. But if your butyrate is produced too much, then you need to reduce the production. And how are you going to do that? You're just going to reduce the substrate that your bacteria are using to produce it.
So that we eat less of certain foods that feed that bacteria.
Exactly. So the computer would automatically say, okay, you have these bacteria that are hyperactive in producing butyrate. We need to find what substrates they're using for butyrate, and let's put those foods into minimize or avoid, depending on how big the problem is, right? And if you have too little butyrate production, which is by far the most common problem in the United States, in developing countries, because we don't consume fiber or enough fiber, then the computer would say, okay, this person has these bacteria and they're, or whatever microorganisms they are, and they've activated these butyrate pathways, but they're starved. They're not reaching their full potential. So now we need to stimulate those pathways. And how are you going to stimulate them? You're going to provide them with the substrate.
Mm-hmm. And how do we, so if it's more common to have too little butyrate, and butyrate, those bacteria are fed by fiber, what are the consequences of having too little butyrate? What are the symptoms?
Well, there are many symptoms. So some of the symptoms some of the things that I really like to bring up are, butyrate literally is the food for our intestinal epithelial cells. So the one cell layer that's lining our intestines, they're called intestinal epithelium. Those cells have evolved over, millions of years to actually use butyrate, that is basically micro poop from bacteria, as food. So they're using it, not glucose, but they're also using glucose, but they're using that butyrate. And so to maintain health of the intestinal lining, you need butyrate. To me, that's probably one of the best, healthy uses of butyrate, but there's many others. The one that I really, really love that I mention a lot is, we all know people who get sick once or they get vaccinated once and they don't get sick again. Mm-hmm. And we know people who repeatedly get sick, whether they get sick from the pathogen whether they get vaccinated or not, right? Mm-hmm. And this phenomenon has been known since the advent of vaccines and since we've known about infectious diseases, that some people respond to vaccines and some people do not. And some people get sick once with something and then they're protected and others don't. Mm-hmm. And just maybe 2 years ago, we teased apart, and when I say we, the scientific community, we didn't discover this at Viome. The scientific community teased apart the mechanism, and I'll now get a little technical, but basically, for viruses, the key immune cell that fights viruses is called T cell. Mm-hmm. T cells will recognize human cells that are infected by the virus and kill that human cell, therefore kill the virus, because the viruses require human cells. And these T cells are cytotoxic T cells, and they're active during the infection. However, after the infection, they will die unless they transform themselves into something called memory T cell. Memory T cells essentially live in your bloodstream for decades. Mm-hmm. They remember that pathogen. That's why they're called memory T cell. They remember that pathogen so that next time you get infected by that pathogen, they jump on, they kill mm-hmm. it before you even have symptoms.
I see. And I would imagine in some people that process happens very readily or easily, and other people maybe not so much.
The trigger for that process is bacterially produced butyrate in the colon.
Really?
They only respond to that molecule. They don't respond
to all transitions from T cells to memory B cells? That's memory a general thing?
Yes.
Wow.
So butyrate is required for that transition. And so if you're not eating fiber towards the end of your illness or right after your illness, when you still have those cytotoxic T cells that are reactive to the pathogen, if you're not eating the right fiber for your gut microbiome, or if you have lost members of your gut microbiome that produce butyrate, then you can eat all the fiber you want. Your immune system will not remember that pathogen, and you will get sick over and over and over again.
Wow.
How crazy is that?
Yeah, that's remarkable, because it's like context dependence on multiple levels. Like, obviously you have to get the infection for the immune system to kick in at all. And then you need to have the right diet induced microbe byproduct at the right phase of that infection to initiate the transition from the one type of T cell to the memory cell.
Yeah, it's crazy.
Wow.
So these are super important things, and for our physiology, and this is just one of thousands of examples where microbiome byproducts influence our mental, physical, cognitive, and immune health.
Yeah. So talk a little bit more about essentially, so you're not measuring just the microbiome population. You're not measuring which species are present and absent. You're doing that, but you're also measuring what we can just think of as microbial behavior, what they're doing, whether or not they're producing certain metabolites or others, and how quickly or how readily they're doing this.
How much of that, because the quantity is very important. And
Mm-hmm.
our method is natively quantitative. And as we discussed earlier, you need to have just the right amount. You can't have too much or too little of almost anything.
Mm-hmm.
When we talk, here's another example: hydrogen sulfide gas. Hydrogen sulfide, many people say, oh, that's bad. That gives you bad gas. And there is definitely scientific literature showing that it causes inflammation, right? Too much sulfide gas. However, people who don't have enough of it, they don't have normal functioning gut. It's involved in bowel movements and in normal healthy gut. And so you have to have just the right amount. So you can't just stop eating anything that has sulfur in it and think you're going to be healthy. So you
What are
need.
examples of foods that have that?
So cruciferous vegetables are best known for having sulfur. And so if you have too much, our computers are probably going to put many of the cruciferous vegetables in the minimize or avoid list. If
Mm.
you don't have enough, the computers are going to say, hey, you need to eat a lot of these.
So that would be
Or
like broccoli, cauliflower, stuff like that.
exactly, Brussels sprouts, cabbage, exactly. Those are all sulfur containing foods. And then you have specific sulfide containing molecules as ingredients for sulforaphane, I think is what it's called, that have biological activity beyond just hydrogen sulfide gas. And so, going back to your question about, people think steak is a steak and broccoli is a broccoli. The way to think about food is that a typical omnivorous diet has about 15,000 different chemicals that we introduce in our body. So after you chew up all the food, like if you take all your diet that you consumed over the previous month and you blend everything and you measure all the chemicals in there, there's going to be about 15,000 chemicals. Some of those are going to get absorbed directly into our body. We do not need the microbes to help us with that. Some of them will get processed by the microbes, and they will then convert those foods into secondary metabolites, what I called micropoop earlier, what we discussed as ethanol and butyrate. And those secondary metabolites, humans have evolved to depend on for health. And it's this flood of chemicals that comes in with the food that interacts with our body and the microbiome that determines, are we healthy or are we not healthy? So it's really that big combination.
Can you walk us through? So walk us through like one of your major products that's giving like the comprehensive picture that's measuring all the different samples that you guys take. Walk us through an example of how this works, just the mechanics of how it works for someone, but then maybe do it. Can you give us sort of like a case study of a typical result that's very common that you see across a lot of people in terms of, something they're deficient in or something they need to cut back on or whatever?
I'll give you some examples, but there is no typical. We have 500,000 customers and we have 500,000 diets. There is no typical anything. So a customer would basically order a kit. And so the one I always recommend is the full body intelligence, the FBI kit. They would receive a box in their home. The box has 3 sample collection kits for stool, blood, and saliva.
Mm-hmm.
Even the blood is collected at home. Of course, saliva and stool are collected at home. So they would collect those 3 samples. I can collect all 3 of those samples in under 10 minutes. Now, I'm the guy who developed this technology, so I've done this hundreds of times, so I'm pretty quick, but it's a pretty quick process. And then they ship these 3 samples in a prepaid mailer directly back to our labs, which is where I work. And we take those samples and we perform the exact same analysis on all 3 samples. And that is, we basically purify RNA molecules from all 3 samples. And those RNA molecules come from every type of microorganism you can imagine and all human cells that are in that sample. And we analyze all RNAs, independent of their source, whether they're coding for a protein or not. And all of those data get uploaded into the cloud. And then we output bioinformatically at the strain level which microorganisms are found and at what abundance. And then what are the genes that the microorganisms are expressing and what functions are they performing? And then for human genes, what is the gene expression level for human genes? All of that bioinformatic information is now used and combined with the questionnaire that we ask our customer. And so our customers will say, these are the symptoms I currently have. So they may have IBS constipation or IBS diarrhea, or they may have depression, or they may have inflammatory bowel disease, or nothing. And then we ask our customer, what is your number one health goal? Mm-hmm. And that can be to reduce depression, that can be to improve lean muscle mass, that can be to lose weight, whatever they say that is. And then we ask our customers to tell us, do they have any food sensitivities or allergies? And they want to avoid any foods for any reason. And those inputs now, combined with all the molecular data the computers take, and their job is to output all foods into 4 categories, to group all foods into 4 categories. The 4 categories are avoid, means you should never eat them. And then 3 other categories that you should eat, and they're minimize, enjoy, and superfoods based on the amount that you should eat.
Mm-hmm. And so superfood, I imagine superfood doesn't mean it's intrinsically a superfood as that term is often used, but it means for you should eat only quite a bit of it.
Exactly. Yeah. Only for you.
So you're basically doing, it sounds like the essence of what you're doing is you're collecting 3 different types of samples and you're doing mass scale transcriptomics, gene expression measurements on all of that.
Yes, exactly. Gene expression analysis. Because we want to look at the activation of the genes, the activity, the biochemical functions, and not just who is there, because just the fact that something is there, you have no idea what it's doing. So there is no other functional gut microbiome test on the market today. They're only telling you who's there, but you have no idea what they're doing. And that's really, I knew this back in 2010 when I sort of created this platform. I was a senior scientist at a genomic facility, and we used 16S analysis, metagenomics analyses, and I knew that that's the fundamental deficiency is just because a microbe is found somewhere, you don't know which of the 4,000 genes that it's encoding are actually functional and to what level. You don't know the behavior.
Yeah, it's like, what would an analogy be? Like if you had 2 cities and they both have 5 million people in them, and they might have the exact same demographic composition, they could have very different economies and cultures because of the way people are behaving and interacting in each place.
Very different, exactly. Very different behavior, exactly. And so that's what took 6 years to develop this technology before we started Viome, because I wanted to have the right data set, the relevant data set for human health. I didn't want to just look around and say, oh, 16S is cheap, it's easily done, it's, there's kits for that, let's just start a company. I spent 6 very long years begging for money and doing research to develop this technology. Okay, so now the customer basically receives these food recommendations. They also receive supplement recommendations, which is which supplements they need and in what amounts. That's what we call results and recommendations. The results include about 70 different molecular scores. These are very much consumerized at this moment, meaning the names are consumerized, but the science under them is 100% science and robust. And we now publish 2 papers on that. So people can actually open the hood and understand exactly what we measured, how we computed everything. Everything is mathematically explained down to the last 4 decimal place for everything.
Mm-hmm. In what instances do you make food recommendations versus supplementation recommendations? Why would you take a supplement versus getting it from a natural food or vice versa?
Yeah, so I don't know all the algorithms, but I know some. And so I talked about sulforaphane, for example, or quercetin, for example. So when a food has micronutrients, that stimulate production of both harmful and beneficial metabolites in your body, there's a conflict. Should the computer recommend that food to you or not? Right? If strawberries have lutein and they have quercetin and they have some other polysaccharides that are not good for you, the computer is conflicted. And so the computer says, okay, one solution is if there's a supplement for the good ingredients, then I can actually minimize or avoid this food, but provide that beneficial ingredient.
So if I have an allergic or a sensitivity to tomato or nightshade or something like that, I can take a supplement to get the nutrients that I want from that thing without exposing myself to the allergen.
Exactly. That's another logical circuit. Yes. So supplements are exactly what it says.
Yep.
They're a supplement to food. And it does not mean that you can just take the supplements and eat any crap you want and expect to feel better, because the foods contain 15,000 supplements in them, right? Every chemical
Yeah.
in foods is potentially bioactive, and it's a supplement. And supplements, if you take 30 to 50 of them at most, that's still a tiny fraction of the chemicals you're getting in foods. Plus, the amount you're getting in foods can be significant. Not always, but can be significant.
Mm. Is a major factor for why our microbiomes have changed over time and why people have certain sensitivities now at much higher rates than they used to? Is it because of not just, so when we say antibiotics, we're usually talking about something you've taken because a doctor gives it to you for the purpose of being antibiotic because you're ill, but also all the preservatives and stuff in the foods effectively are antibiotics. They're anti-microbe molecules. Has
It's a
that stuff
different
just
name.
has that stuff just like depleted people's microbiomes over time?
Well, so there are several factors. That's one of them. So we have customers who no longer possess microbes to perform a certain function. For
Yeah.
example, we have customers who have lost the ability to process oxalates.
Yeah.
And so until the industry provides probiotics, live probiotics that can actually consume oxalates, which I understand some companies are working on oxalobacter and maybe other bacteria. Until there's such a probiotic, unless you're willing to do a fecal microbiota transplant to reintroduce that microbe, you lost that physiological function. You don't have it. If you've lost major members of your gut microbiome that produce butyrate, even if you have minor ones, those minor ones may not be able to produce enough butyrate for you to be a healthy human being, right?
Mm-hmm.
And this goes on and on and on. And so that is a major problem. The second major problem is that we used to not move around, and we used to basically co-evolve with the microbes that lived in that environment and with the food that lived in that environment,
I would
right?
grow up and live my entire life in one region.
In one region, and then your mother, when she gives birth, would pass those bacteria to you, and you would pick up the rest of them from the
Yeah.
environment. And
And
so
you
there was
wouldn't
that
be,
seeding, continuous seeding.
yeah, and you would be eating the traditional foods of the region. You wouldn't be importing stuff from all over the place and around the world, et cetera, et cetera.
Yeah, and now we've lost that touch with nature
Mm-hmm.
from birth. Literally at birth, 30% of the kids are born now with C-section, meaning no transfer of gut microbiome. Second, everything is sterile at birth and since birth, right? Your office is sterile, your kitchen is sterile, your food is sterile, your everything is sterile these days. Whereas if you just look at the life 300 or 400 years ago, there were no soaps, there was no toilet paper, there was just no washing and peeling and sterilizing of fruits and vegetables. You just pick a fruit or vegetable and you eat it, right? If you dig up a carrot, you just ate it.
Mm-hmm.
You didn't peel it. And now we buy baby carrots all highly processed with no microorganisms. And so we lost the ability to reseed or rewild our microbiome from birth.
Hmm.
We started using antibiotics, and antibiotics are not just in pills that you take from the doctor, but they are found in many foods. So I think most people understand that the major antibiotic use is in animals. And so when you eat non-organic meat, you're likely consuming antibiotics, and the food
Yeah.
industry finds very clever ways to sneak them in no matter what. One particular example that I want to point out is when you see all natural label on meats, okay, the definition of all natural means that from the moment the animal dies, they did not add anything to that meat. But until the animal dies, they can feed that animal any amount of antibiotics, and that's okay. You can still call it all natural. And so that tricks people into thinking they're consuming healthy food, right?
Mm-hmm.
So antibiotics found in foods, and then preservatives. You have your sodium metabisulfite, you have your potassium sorbate, and others, and then you have detergents. Polysorbate 80, for example. If you put creamer in your coffee, that creamer likely has a detergent in it to prevent it from separating. And so they put detergents in so many foods to prevent separation of fats from water. But these detergents can strip your gut lining and cause a leaky gut and cause all kinds of immune reactions. And so those are all examples of why we have this huge mixture where people no longer have the fully wild microbiome that's adapted to their environment. Yeah. And so now we have to find very creative ways to recommend foods to those people. And still, with some people, like the example I gave you, when we find no oxalate pathways active,
Mhm.
and we find that that person has no microorganism that even encodes oxalate processing genes, there's nothing you can do about it. You basically cannot cannot consume much oxalates basically for the rest of your life unless you
Hmm.
somehow acquire that microorganism, which is unlikely in today's sterile life.
Interesting. And so that kind of brings me to another question I wanted to ask you. What is your general take on using dietary changes to facilitate changes in your microbiome versus taking things like probiotics that contain specific species?
Well, those two are complementary, and I think that many people have plenty of lactobacilli and plenty of bifidobacteria and plenty of streptococcus, and so they don't really have a need for much of those. Um, and you can just feed those existing microorganisms and they'll be there for you. If you lack some of those, you can reintroduce them with those probiotics that are available. Now we even have live Akkermansia as a probiotic and some other ones. And I hope that more and more companies bring more and more strains of probiotics with more functions to the market so that people can rewild their microbiome. I am very certain that one day we will know all of the microbial functions that are needed for our healthy body, and that all of the probiotics that have those functions will be available commercially, so that at any given time in your life, you can always at least rewild your microbiome to the point where it has all necessary components for healthy life. And then you just have to feed it the specific foods that your microbiome needs in order to produce those beneficial chemicals, but not produce harmful chemicals. And that's really the trick. It's the contrast between, the microbes don't have a clue as to what's good for you and what's bad for you, and they don't care about what's good for you or what's bad for you as long as it doesn't kill you tomorrow, right? They just care about what is energetically the most favorable food for them. Just like if you put a kid to a buffet, they're not going to choose what you want them to choose. They're going to choose what they want to choose, just like that. And so the bacteria will take what's best for them and they'll make secondary metabolites out of that. And it's just a matter of us now modulating all those functions via foods. Whether you're consuming probiotics or not, whether you have a lot of them or not, it's still the same problem.
Mhm. And yeah, you guys do a lot of research. I know that you've done some clinical work. What are some of the ailments that you've been able to successfully improve for people through this general type of approach, and how rigorously have you demonstrated that?
Well, I would encourage people to read our papers. We have a whole bunch of papers that we've published, and that includes some efficacy papers. If people just go to viome.com/publications, all the papers are there, but we have papers that disclose our entire platform from start to scratch, meaning from start to end, meaning how do we actually clinically validate our tests, stool, blood, and saliva? What kind of molecules are we measuring? How are we measuring them? And validating all that because this is a clinical test. Then we have the entire process from start to end in terms of food recommendations. And then we disclose, like I said, how do we create these scores, molecular scores that are visible to the customers as results. And then we have two papers published on efficacy. So we have one paper that we published in 2023 in the American Journal of Lifestyle Medicine, where we used some clinical measures for depression, anxiety, IBS, and type 2 diabetes, showing the efficacy of our nutrition. We also just published a preprint last year for efficacy for HbA1c, which is a major biomarker of type 2 diabetes, in a randomized controlled trial. So we're starting to do more and more clinical trials. We used to heavily focus on clinical studies where we learned, and now we're doing those, and we're now doing randomized controlled trials to see how well this works, to test.
How do you guys? So one of the things, I think we touched on it maybe a little bit in the beginning, but one of the challenges for scientists and for anyone else who's just interested in learning from the literature is that the scientific literature today is so vast and contains so many studies of variable quality that have conflicts of interest sometimes disclosed, sometimes undisclosed, that are, sometimes the results are very good and sometimes they're not very good. It can be difficult to discern all of those things. How do you guys grapple with that problem, especially when I'm thinking about the ways that you probably have to integrate the literature and all of that information into your algorithm design?
Yeah, so our major algorithms are actually designed based on super solid literature and our own data. Uh, and so if you look at, for example, the paper that I mentioned in 2023 on our oral microbiome scores, you will see that we used 20 something thousand people for that study. And if you look at the paper that we published last year, which is a preprint for inflammatory bowel disease and scores related to that, we used 60 or 70,000 people. So we perform extraordinarily large studies on humans to figure all this physiology out. And then we do go back to the literature and find very solid evidence. And so when we talk about things like butyrate, for example, when we talk about lipopolysaccharide, that evidence is not really conflicting. That evidence is now rock solid. That also when it comes to hydrogen sulfide and ammonia production, and there are just so many of these physiological aspects that are not really speculative anymore. There are many that are still speculative, but the evidence is strong enough that we will take those into account. But now we're talking about edge cases for our platform. Our platform now is so mature that the vast majority of it is based on very, very solid evidence. And there are some cases where it's not, but those are there's still good evidence. So yeah, and as the world knows more and more about things, then when we talk about, for example, trimethylamine and trimethylamine oxide literature is very, very homogeneous in terms of TMAO is bad for cardiovascular health. When we talk about blood glucose, high blood glucose is generally very bad for your health. And so those are the kinds of things that I think are perfectly fine to adopt. And when it comes to blood sugar, for example, we ran a multi-million-dollar study in the USA and Japan that consumed half of our company for a year or more, where we developed a very objective machine learned model of how high your blood sugar is going to spike when you eat different foods. And so these are the kinds of things that we take very seriously. The literature was not very good at that for that particular condition, and that condition is very prevalent in the United States. So we paid special attention to that. The same with IBS. There's very conflicting literature about IBS because it's a heterogeneous disease. And so we've spent a lot of time and money analyzing the data for IBS, same with IBD and so on. So, it's a complex answer, but basically we've done the best we can with what's available, and it's going to get better from here.
How much do your most commonly used products cost today? And what does the price trajectory look like? Are costs coming down over time due to technology improvements?
Yeah, so we started at $1,000 for one test, and now I think it's like $300 for three tests. I think, to be honest, I don't track the prices, but it's somewhere in the order of $300 or $400 for three tests. But the absolute goal of mine, and I will not stop as long as I'm alive, is for the entire Viome platform to be completely free to the user. That is our end goal. Our end goal is not to make it available to wealthy people. It's to make it available for free to everyone. And how's that going to work? The moment that we have very solid mathematical models that are health economic models, where we say, whoever is paying for your health, if they invest in Viome, in your lifetime, they're going to spend this much money. If they don't invest in Viome, on average, they're going to spend this much money. And as long as spending on Viome is less than what they would spend for your healthcare, they're going to pay for Viome. So whoever is the payer, and the payer today can be a vertically integrated insurance and provider company. It can be self-insured employer, which is very common in the United States. And it can be a national healthcare system like in the UK, Canada, Australia, and many other countries, right? Whoever is paying for your healthcare, they're going to want to pay for Viome and give it to you for free. So that's really where we're headed.
Mhm. Um, what about data security and privacy and all that stuff in a world where we've got 23andMe data breaches and
Yeah.
Facebook and all
Yeah.
that? How do you guys approach that?
Yeah, we approach it very seriously. We are a data science company, so we depend on the data. Um, I can just tell you that um, we have two completely separate databases. One is identified, which is just a basic information, like super basic address, name. Um, and that information is tied to a barcode. And then we have a big database with all of our molecular data and all of our phenotype data, and that's tied to that barcode. So it's completely de-identified. So essentially 99.9999% of our data is all de-identified. It's just complete gibberish. You would have no idea whose they are. And those two databases are completely separated by physical and access and authorization and two-factor authentication that's separate for those two. And access is granted only for the people who need to have access. Um, so that's one. So we follow the HIPAA security protocols. So that's sort of the highest grade that you can. And I think we've gone beyond that as well. And then last but not least, remember that we're not doing genetics. We're not doing DNA sequencing where you can unambiguously identify someone. We're doing gene expression. And your gene expression changes every 6 months. And so even if someone hacks your gene expression data from 6 months ago, that was you 6 months ago. You today is different from you 6 months ago. And so it's a very dynamic sort of a system that we're measuring. We're measuring the physiology. We're not measuring the DNA. We degrade DNA. The moment every single sample that comes into our facility, the first thing we do is we extract RNA and DNA, and then we degrade DNA. And so we don't analyze DNA at all.
Mm-hmm. And so is this, so you mentioned like every 6 months, is that approximately the time scale at which people will plausibly see meaningful changes in their microbiome just from the normal sort of drift of life?
Yeah. So we did a study where we took something like 1,200 customers and we showed that in a statistically significant matter, so p-value went below 0.05 in terms of is the microbiome different from 6 months ago, right?
Mm-hmm.
It takes 6 months to change it in a statistically significant matter. That is the average. There are people like me who are very stable. My microbiome hasn't changed statistically significantly overall in 3 years.
Mm-hmm.
But as we discussed earlier, my oxalate metabolism has. And so how do you define change? It's very complex, but overall, it changes on average every 6 months, which is why I think that eventually when Viome is free for everyone, every 6 months we will be testing everyone on the planet.
Mm-hmm. And I guess sort of at a very high level, the idea here is that because these things allow for preventative actions to be taken, if a product like this can become widely available at ultra low cost, that's ultimately going to be cost effective in the aggregate because there will be fewer healthcare costs for people over the course of their lifetime.
Exactly. I mean, if you can prevent IBD, arthritis, Alzheimer's, cancer, diabetes, I mean, those are $4.9 trillion, 18 or 20% of our national gross budget is dedicated now to healthcare. And in some countries like the UK, the healthcare costs have gone beyond what the country can actually handle
Mm-hmm.
to the point where people are saying that they cannot be seen by a physician for a year or two or three,
Mm-hmm.
and so these and they keep increasing.
Mm-hmm.
There's no sign of leveling off. And so we as a society have to focus on preventive healthcare, but we have to find a way for it to work as a business model, because if we don't, then it's not going to work.
Mm-hmm. Mm-hmm. Are there examples where you've uncovered so many people are taking so many pharmaceutical medications for all kinds of chronic illnesses at this point? Are there any examples out there where, like, say there's a diet induced change in the microbiome and someone's got some deficit that results in a symptom and they get diagnosed with something and prescribed a drug, and that's done with the thinking that that's some intrinsic problem with them that they just have to live with forever, but in fact, unbeknownst to them or even their doctor, there is a sort of diet or microbiome thing that could easily be fixed if they only knew about it? And that would get them off their medication.
Well, unless someone has a inherited genetic disease,
Mm-hmm.
the answer to all other cases is yes. You
Mm-hmm.
can prevent all other diseases just by physiological tuning. That is why the disease burden was not so high 100 years ago, and now it's so high, right? It's not because of genetics. So yeah, the answer to that is exactly why are those people on Greek islands sheep herding and farming at the age of 90 and having a normal conversation and not getting sick, but they've never seen a doctor or taken a pharmaceutical in their entire life because nature has already figured all this out.
What what do you think? So is this something that you think people should just do like once a year? Just treat it like they would think about a normal yearly checkup or normal blood work. They would get something like this, and every year they're probably going to find out something that might need to be changed.
Well, I recommend everyone who can afford it right now to do every 6 months. I also recommend everyone who can afford to do a 96 food sensitivity test. I think that that test and our test, full body intelligence, are going to cover the vast majority of biomarkers that are important for our health related to nutrition and lifestyle. And then if you have symptoms, you can go beyond that. You can go into testing for heavy metals, you can go into hormones, you can go into mycotoxins and other more exotic tests. But these two should be the basics every 6 months.
Mm-hmm. And what kind of how is the data made available to consumers? What kind of interface is there? What does the UX look like? And can they really see everything that you guys measure?
I mean, we display results and recommendations that we validated clinically and that our CLIA license allows us to display. And though that's a dynamic, over time it'll change. But what's there is there, and that can be seen today on both mobile and web applications. We are currently in the process of completely revamping our app. So in 3 months, it'll look completely different from today. But the basic output is exactly the same. And that is, here's the profile of your physiology. We have 70 different molecular scores that we put together. Here's where every single one of them is relative to a healthy population. And then what are the 4 food categories? Place all foods into those 4 categories. And what are the supplements you need to be taking in what amounts? That's basically it. That's what I will always be extracting from Viome, independent of what the interface looks like. Like
Mm-hmm.
I don't care what the interface looks like, basically. I'm not going to go digging about, all the text. There's just so much content to browse through.
Mm-hmm.
But to me, right now, literally just download my recommendations, put them into ChatGPT, and ask it to make me a meal plan with certain daily calories and certain amount of protein and certain restrictions.
Mm-hmm.
It's that simple to me. That's really the
Yeah.
practical use of all this information.
And if someone gets this every 6 months, 12 months, whatever, do you track and display the historical data so you can see how things have changed over time?
We do. Yes, we do.
And what about the is the raw data itself exportable and savable? Can I save my data and dig into it on my own?
And how do you define raw data?
I mean, just the underlying measurements that you guys make to make all of your
Yeah,
recommendations.
everything that is clinically validated under the CLIA license is displayed on the app. Um, Results, I honestly don't know if results are downloadable. You can take screenshots, you can export as PDF. I'm not really sure.
Mhm.
But I know the food recommendations are downloadable because I've done that.
And so what are you guys developing right now? Are there whole new types of tests that do things that are not available at all today in any way, shape, or form? Or are you focused more on just getting more granular with all the microbes and all of the gene expression stuff that you measure?
All of the above and much more, everything that we possibly can. Yeah, moving the platform as fast as we can in many, many directions. And, every 2 to 6 months, we change the focus of the company. Like right now, the focus is on improve some database and improve the app in a big way. But we also worked on some additional tests that we may release this year or next year. And we are always improving. When I say improving the database, meaning that we're actually adding more foods to our database, including some ethnic foods that are not so common in the United States, but still there's enough immigrants that consume them, and also for the foreign market. And we're also always improving the database in terms of molecular interpretation of the data, because those are always growing. We're always learning about new microorganisms and their functions. We depend heavily on the KEGG orthology database, the KEGG database. And so all of these things, there's just so much that it's just like a giant platform that has many moving pieces, and we're trying to move as many of them as fast as we can.
What would you say that in your view, many of the illnesses that chronic illnesses that people are experiencing today are treatable in very simple ways, just by changing diet and lifestyle and things like that? Are there many things out there where like people treat them like terminal illnesses or something that can't be fixed, where it's not necessarily a genetic or intrinsic problem that they have that they're stuck with forever, but it's something that can be fixed through a relatively simple set of lifestyle changes?
I mean, relatively simple. That's hard to state. It's a complex topic, but I would say that for the vast majority of cancers and chronic diseases, we are going to learn in the next 5 to 50 years, we're going to learn how to prevent them.
Mm-hmm.
If the example I like to give is the Black Death. So in the 13th and 14th century, a third of the world population died from Yersinia pestis infection, right? Called the plague or the Black Death. And at that time, the level of science was essentially what it is today for chronic diseases and cancers, meaning you either died or you didn't. And
Mm-hmm.
if you didn't, hey, good for you. You must have some good luck. And if you died, whoops, bad luck, right? Today, people are falling like flies, right? Someone has multiple sclerosis, boom. Someone has Alzheimer's, boom. Someone has ALS, boom. Someone gets cancer, boom, right? Bad luck. But none of that is bad luck. Not a single disease or cancer is bad luck. It's just biochemistry that we do not yet understand. And once we can understand it, just like we understand the infection that Yersinia pestis causes, today, no one wakes up and if I ask you, will you get the plague tomorrow? You can confidently answer, you will not get the plague tomorrow. You know that because you know where this pathogen lives, you know how it's contracted, you know how it's treated, all that stuff. So you're not worried about it. But if I ask you, will you develop pancreatic cancer? You have no idea. It's completely bad luck or good luck, right? But in fact, there is a series of biochemical reactions in our body that leads to pancreatic cancer. And once we figure out what is that cascade, we will be able to prevent it. We just have to do it. But it's not as simple as it sounds. If we all as a society organized ourselves to do it, we would absolutely solve the problem. We don't need any new technologies. We already have all the science and technology to do it. We just have to reorganize our scientific community in a way that's focused on solving these problems versus let's just study things and publish papers and study more things and publish papers, right? And so, yes, let's do it.
Are there any final thoughts you want to leave people with, or anything you want to reiterate based on what we talked about in terms of what you guys are doing, or just the concept of systems biology and preventative medicine?
Yeah, we can do some takeaways, sort of summary, and then people can review the summary, and then if they're interested, they can go back and listen to all the details. So, what are some important things, takeaways that I would say? First of all, is that our health and diseases are not determined by genetics alone, or immune system alone, or foods alone, or microbiome alone, or hormones alone, or heavy metals alone. All of these things combine to produce either a healthy body or a sick body. And so almost every disease and cancer are multifactorial. That's one takeaway, and that's where this concept of systems biology comes in. The next concept I would like people to think about is that we no longer live with nature. We're not a part of nature anymore. We've extracted ourselves from nature. when we lived with nature, we basically consumed specific foods from that environment. We lived or co-evolved with microbes that lived in that environment, and everything was in sync basically, and that maintained our health. Today, we have broken that symbiosis, and we have broken that homeostasis, that healthy homeostasis, by consuming too many antibiotics, either as pills or found in meats, consuming too many preservatives, food additives, by living a clean life. Purell and Clorox wipes are everywhere. We don't go out in nature. People don't go out and get dirty. People don't eat with their hands. People don't do organic gardening. Everything is peeled and washed and sterilized, all the foods, right? So we really need to go back to nature and live with nature. You can partially, and depends on the person, restore some of that via probiotics. But for some people who've lost members of their microbiome that are not found in the probiotics on the shelf, they can't restore those yet. So that's the challenge. So the takeaway there is go play in nature more, eat more organic food that has soil on it. Don't sterilize everything. Choose foods that have no additives, just whole foods. Go buy whole foods the way you see it at the grocery store is the way that it came off a tree, and eat that, or meats that are, well raised. I guess this, grass fed is better than corn fed meats. So that's a big takeaway. Another one is that if you're going to make any changes to your diet or any part of your lifestyle, you really need to be testing and to understand what is wrong with your physiology. And the tests that I recommend to start with, if you can afford them, would be the Viome's full body intelligence test, which is saliva, stool, and blood, plus the 96 food sensitivity test from Everlywell, and only that company. I don't trust any other tests for food sensitivities, and I know that there are some that are complete scams. Mm-hmm. Others I haven't really checked. They may or may not be scammers, but I know that Everlywell is running a clinical test that's very robust.
And that's separate from you guys at Viome?
Yeah, we have not. They're perceived by many people that they're a competitor of ours, but I don't see it that way. I think their test is very complementary to us. Food sensitivity test measures direct immune system responses to specific foods, mm-hmm. Which we do not measure. And so you really want to have both of those. And I've used those tests combined with our Viome test for my family for many years, and I'm super happy with that. And then if you're happy with those two tests, or you don't have any symptoms, then just every 6 to 12 months, whenever you can afford it, run those and follow those recommendations. And definitely pay attention to the recommendations from Viome that give you sort of a general idea of what to avoid, like deep fried foods and breaded foods and processed food, ultra high processed foods and all that. Then if you still have symptoms, you may want to go into hormone testing, heavy metal testing, and mycotoxin testing, because those may provide you with the answers that Viome and Everlywell did not, or complement those very well. And those are also very actionable. And can help you relieve some symptoms. And so test, and then experiment, and then retest. So that's one takeaway. Another takeaway that we didn't actually discuss a lot, that I am super interested in and super focused on, is we are foodies. My wife and I are foodies. We cook all the time. We love to cook. And so I would recommend to every listener to try to do as many cooked foods at home as they possibly can. Just absolutely minimize going out and eating anything, because there are very few restaurants that actually offer whole foods for mm-hmm. food. It's all ultra processed. Who knows what chemicals they added? You're just risking accumulation of some kind of physiological anomalies over time, or risking IBS or food poisoning or any combination of others. Cook foods at home. Go buy whole foods at the grocery store and cook them, and find a way to make it fun. I mean, I know that's easier said than done, because I have friends who just don't like to cook, which to me, that's incomprehensible, because to me, cooking is one of the funnest things in life. So it's hard for me to understand how someone cannot like cooking. Figure out for yourself, what is it? Is it Is it the planning? Like, I don't know how to plan a meal. Go to ChatGPT. You can literally prompt ChatGPT by saying, uh, get me a recipe that is Thai food with chicken or tofu or whatever you want. And I want to make it for 4 people, make it medium spicy, avoid broccoli and spinach, and make sure that it has 25 grams of protein per serving and that it has maximum of 500 calories per serving. And ChatGPT will come out with that. And then you can say, make it vegan or make it whatever you want, right? And so utilize the tools you have if that's the problem, the recipes. If the problem is going to the grocery store, I don't know, there's too many circumstances, but figure out a way. If the problem is food prep, for example, you come home at 6 p.m. and you're like, I'm too tired to chop vegetables and wash them and all that stuff. Okay, then carve out half a Sunday, go buy a bunch of vegetables, wash them, chop them, involve the whole family. It can be a fun family thing. That's what we've been doing for 20 years. And then freeze them. And now you have packets of mushrooms, onions, bell peppers. Everything is frozen, literally ready to go. I can go into my freezer on a day that I'm busy, that I come home late. I can pop in onions, garlic from the fridge, mushrooms, broccoli, green beans, throw them in the pan, throw some cut tofu, add some spices, whether it's Japanese miso or Thai curry or Indian spices. And by the time the rice cooks on the rice cooker, the dinner is served. It's just a no-brainer, and it's healthy, it's wholesome, it's super good. So I encourage people to cook more at home and to involve the whole family and just identify those barriers to that and then overcome those barriers somehow. I don't know what else to say there, but that's a big one. That's a big one because even if you do all this testing and even if you understand how important nutrition is, if you don't have a way to providing that nutrition to your body, well, then it doesn't matter. All right, I'll stop talking for now.
All right. Well, we covered a lot. This is cool stuff. And these products are interesting. It's nice to talk to someone where people can go out and try these things and get some data. And it's not purely just an intellectual exercise for us. But fascinating stuff. Thank you for sharing. And thank you for your time.
Of course, my pleasure. Thank you, Nick.